Dear students, towards predicting the protein structure, the first step is to predict the secondary structure of proteins from their primary sequence or the primary structure. Of course, once you have arrived at the secondary structure, then you can predict the tertiary structure or later the quaternary structure. So since this is the first step in predicting the protein structure, I'll be talking about predicting the secondary structure first. So since the first step in protein folding is the formation of the secondary structures, it is quite useful to see which amino acid prefers to get into which type of the secondary structure. We know that there are 20 different amino acids and that there are about 4 different secondary structures that are formed. These secondary structures include the alpha helices, beta sheets, loops, and coils. So by looking at which amino acid from each of these 20 amino acids prefers to form an alpha helix or a beta sheet or a loop or a coil, then we can kind of guess where an amino acid will prefer to exist within the secondary structure. So by looking at the PDB, which contains all the structures that are known for the proteins, now we know that alanines are very good helix formers. Most of the times, alanines exist in the helices. So if you have lots of alpha helices in a protein structure, then you can guess that there must be a lot of alanines. Or vice versa, if you have lots of alanines in the primary sequence, then you can kind of guess that maybe these alanines are going to form an alpha helix. Now, what if we run such a survey on the entire PDB and we see which amino acid exists in which secondary structure in how many numbers? So such a statistic can be extremely useful in predicting the secondary structure folding of the primary structure or the sequence. In this table, I show you such a statistic wherein alanine has been shown to have a very high propensity for forming an alpha helix. Secondly, you can also see methionine, which has a very high propensity for formation of an alpha helix, and glutamine has a similar tendency as well. However, if you look at these amino acids, that is valine, isoleucine, tyrosine, then they have a very high propensity to form beta sheets. And similarly, for these amino acids, they exist most of the times in the terns. By looking at this table, you can also see that there are times when alanine may actually be present in beta helices and the terns as well. However, the propensity of that happening is very low. So one can then generalize that by such a propensity table, you can only guess and you do not have a certain answer to if a specific amino acid exists within a specific secondary structure. However, extending the concept, if you have lots of these amino acids within the primary sequence, then the propensity of formation of alpha helix will obviously be higher than the propensity of formation of a beta sheet or a tern. So in conclusion, there are several such algorithms that mine the statistics that are there in the PDB, and they can predict the secondary structures by looking at the primary sequence. The first such algorithm was the Chow-Fasman algorithm that was reported in 1974, and we will see a lot of Chow-Fasman in the upcoming modules.